Okay, so we're about to do the lab assignment for Chapter 4, and I have everything gathered that we're going to need. Salt, powdered meat tenderizer, Palmolive, that was the recommended soap, beakers, pipettes, and the tubes, distilled water, instructions and lab report, and then I also have the rubbing alcohol in the freezer. In our procedures, step 1 is to make the lysis buffer and the Protease solution. I went ahead and labeled the beakers so we don't get them mixed up. And I'm going to make the solution. Okay, for the lysis buffer, it consists of distilled water, the dishwashing detergent, and salt. And I will add all the ingredients. So here's the buffer before I mix everything. I'll mix it and then show you again. So here's it all mixed. Oh. Starkey thinks it's food. What? It's not food, honey. Sorry. So that's it mixed. I'll make the Protease solution next. Okay, for the Protease solution, all you're going to need is the distilled water and then the powdered meat tenderizer. This is the brand I bought. I bought it at Walmart. It's like $3 for this. I will measure everything out and be back. Okay, here's the Protease solution. Stark, stop. Here's the Protease solution. The meat tenderizer dissolved much more quicker than the salt did. So we're going to go to step 2. So for steps 2 and 3, you're going to need 3 milliliters of water, a very small amount, and your tube. You're going to chew your cheeks, the inside of your cheeks, for 30 seconds, then rinse with the 3 milliliters of water for 30 seconds, and then spit the water, don't swallow, into your tube. Okay, now you should have your water and cheek cell solution. A little gross, but you know. You're going to take a clean pipette, and then you put 2 milliliters of the lysis buffer into here. And then invert slowly 5 times. Don't shake. Okay, there's that. Our next step is step 4. We're going to be removing proteins. So we're going to be using the Protease solution next. Then you're going to leave it, after you invert it 5 times, you're going to leave it sit at room temperature for 10 minutes. Be back after that. Okay, it's supposed to sit here for 10 minutes, undisturbed, at room temperature. Okay, it's been 10 minutes, so now we're going to add up to 10 milliliters of cold alcohol. You're going to want to tilt it at a 45-degree angle as you add. Okay, so I added 10 milliliters. You can barely see the solution at the top, but you can definitely see the 2 layers. Ooh, focus. There's the 2 layers. So then we leave this sitting upright like this in a cup. Let me put that in here. I'll just lay it like that. And we leave it for 5 minutes, paying attention to the layers. Okay, so it has been 5 minutes. It's sitting here. And you can definitely see it looks like there's little strings from the cell area, and it's being pulled up into the layer of cold alcohol. I think that's really neat. Okay, so we're going to invert it 5 times, and I'll be right back. Okay, I inverted it the 5 times. You can barely see, but the white stringy stuff is my cheek cell DNA. Looking closely, you can't see it on here. Come on. But there is a small black hair in there. And that could be from any number of these things, but I think that's really cool. My husband gets off work soon, so come on, focus. I think I'm going to do this again with him because he'll enjoy it. What a fun lab.